Alexis McAleer with Exam Mini Video 5, Prompt 2. A protein with 4 amino acids in length forms the DNA sequence below in vivo. This occurs after mRNA transcription. 2 strands are present, a template strand and a coding strand. Which is which? The bottom strand gets flipped, and both of the strands have a start codon of AUG, and the highlighted blue demonstrates that. The coding strand has a stop codon that is 4 codons after the start codon, ATG, in this particular strand. The U becomes a T in mRNA transcription, and the UTG on the top strand is not the stop codon, so it cannot be the coding strand. Therefore, the bottom strand is the coding strand, and the top is the template strand. UAA is the stop codon present in these sequences. UAA, which is our stop codon, allows us to see our coding strand that we can transcribe for the mRNA sequence. All we do for the transcription is change all the T's to U's. After the mRNA transcription strand is complete, we can code the amino acid sequences present. The beginning of the reading frame is AUG, which is code for methionine. Next, GUC, which is code for valine. Next, AGC, which is code for serine. And then AAG, which is code for lysine. Then our stop codon is present, UAA. A silent mutation is shown here where a mutation changed a G-guanine to A-adenine in the coding strand, which replicates to the mRNA strand. The amino sequence does not change because both AAG and AAA are code for lysine, hence why it is referred to as a silent mutation, because most go unknown because there are no effects. Here is a nonsense mutation where a transversion occurs on the first nucleotide. This changes an adenine to a thymine, an A to a T, which then codes for a uracil during mRNA transcription. Notice how the template strand goes unaffected here. The lysine codon is now converted into a stop codon due to the mutation. This sequence now ends early, which makes it a non-functional protein. Here is a missense mutation due to change in one nucleotide in the whole sequence. Where transversion occurs over the guanine, changing it to cytosine, the amino acid sequence does change due to AAC now coding for asparagine and not lysine. Missense mutations have a possibility of going unnoticed or noticed by changing the functionality of the proteins in question. A read-through mutation occurs when a transversion happens with a stop codon nucleotide. An A, adenine, is changed to a T, thymine, an mRNA transcription coding strand. This now becomes a uracil amino acid. Results from a read-through mutation are drastic. The codons used to code for a stop codon are now another amino acid. There is no longer a stop codon present, and the codons continue to code for amino acid, which results in a longer than normal sequence strand. This is an example of a non-functional protein.